Good. So my name is Lisa. I'm a postdoc at the group of Masha Binder for 2 years now, and today I want to talk about immunosequencing in COVID-19 and what it can teach us about responses of B and T cells in this disease. First of all, let me introduce my lab. These are my very talented co-workers who all contributed to the data I'm presenting today. We are located in Halle, which is like an hour south of Berlin, and it's a very idyllic small city. And we are located at the university hospital there. And our group is hemato-oncologists, so we are working on types of lymphoma, both B and T cell lymphoma, for example, DLBCL or NLPHL. And we also do translational research for a number of clinical trials on solid tumors, where we often have two sequencing approaches. We do targeted panel sequencing to identify driver mutations and to track tumor load in liquid biopsy. Yeah, our focus is the immunosequencing on B and T cells. And today I'm going to show you what we did on COVID-19, which is our newest field of interest. Okay, this is our cohort of COVID patients. It comprised mainly patients from our own hospital, where we got samples from active disease time points, but we also recruited recovered patients, which usually refer to the milder disease courses. And so we could get samples from active state and from recovered state and from mild and also severe disease courses. We work with genomic DNA and amplify IGH and TRBV loci with Biomed2 urocanality primer pools. And our sequencing platform is the MySeq Illumina, and we use the MyXCR platform to analyze the data afterwards. We extract only productive clonal types with a minimum of 2 reads and normalize every repertoire to the lowest read count. The first thing we do after sequencing a new batch of samples is to look at the repertoire metrics, clonality, diversity, and richness. And the most important thing is to have a control group, which is also age matched, because we have shown before that age does have an influence on the repertoire metrics, and with increasing age, the richness decreases. And so we are lucky to have a large pool of healthy donors from which we extract an age match control for all our samples. So the repertoires of the recovered COVID-19 patients are richer and more diverse, which is especially pronounced for T cells. And in B cells, we also check the somatic hypermutation. And this was very interesting because the overall somatic hypermutation in average was not different in COVID patients than from healthy donors. However, the patients with active disease, they were somewhat subdivided into 2 groups, and one subgroup had especially high percentage of somatic hypermutation. So we looked closer into that and could correlate the severity of disease with the rate of somatic hypermutation, because the cases with the high somatic hypermutations, they were all ventilated, and the mild disease courses, they had like rather low rates, which is in line with the finding that the COVID-19 triggers especially naive responses. And from the B cell repertoire, we wanted to infer converging clones in order to identify SARS-CoV-2 reactive B cells from patient repertoires. At first, we used the tool Brillia, which infers clonal lineage trees. And here we see that the antibody positive samples of COVID-19, they had more connected sequences. So there was some antigen-specific proliferation going on and some clonal evolution going on. These are the controls where only a few connected sequences are seen. However, as I said, one observation in COVID-19 was that neutralizing B cells are often more naive, and therefore we explored also another approach in creating a phylogenetic tree, this time based on sequence similarity. We did that with FastTree-MP. In this tree, there are repertoires of COVID-19 patients with antibody positive versus antibody negative results in ELISA against SARS-CoV-2. And to get an impression what to expect, we used the negative control two cohorts of healthy donors against each other, and as a positive control, an Ebola cohort, a cohort which was vaccinated against Ebola. And in the two healthy controls, there were like only a little clustered, like clusters of groups together, and in the vaccination cohorts, there were more clusters of the vaccinated samples versus the controls. So when we look back at our COVID tree, we find even more clusters of the antibody positive samples, and especially there was a drift towards sequences using IGH gene family. We wanted to look closer into this very unique response, and this time we wanted to identify protective PCR sequences. So we divided our cohort according to disease severity into fatal and non-fatal, and constructed a tree similar to the one before. We derived sequences from the patches of converging sequences from the non-fatal disease curves, and this was our sequence set 1. Then we wanted to search for these sequences in healthy repertoires and other cancer repertoires from pre-pandemic samples to correlate the finding of these sequences with risk groups. We also used a set of validated sequences from the COVID antibody database, and from that we extracted 2 sets, one with non-neutralizing antibodies and one set with neutralizing antibodies. To explore protein similarity, we analyzed the Kidera factor profile of set 1 to 3 sequences, and these factors describe biophysical properties of amino acid sequences. The principal component analysis shows that all 3 sets differed significantly from the healthy controls. But if we put those 3 sets together, they show a lot of overlap, and they do not significantly differ from each other according to the PCA. So this is really remarkable because it shows that the 3 sets have similar properties, and it validates our derived sequences and the approach to get those sequences, and it indicates that it could be like relevant clones in this set. Okay, so as I said before, one feature of SARS-CoV-2 is that it evokes naive responses, and this brought us to search for these for sequences with a similar rearrangement, because if they are naive, then they could be present already before the antigen contact, which could determine like the response to the virus to be a mild or a severe disease. And we examined that by searching our set 1 to 3 sequences in pre-pandemic samples, and we had 200 healthy donors, which we subdivided into young and old individuals at a cutoff of 60 years. We also had 500 repertoires from cancer patients, which we subdivided into blood cancer and solid cancer. And we searched the sequences for identical CDR3 amino acid sequence within a Levenshtein distance of 2 and identical V and J genes. So these are 2 ways of plotting the data. One panel shows the percent of repertoires which had at least one of the sequences in each set, and the second line shows the mean number of sequences found per repertoire. And you see that, you see that in the COVID group, there are a lot more sequences found than in the pre-pandemic samples, and also they are more found in the active group. Within the healthy group, the sequences were more frequent in the younger individuals than in the older individuals, and in the cancer samples, they were even less frequent. And for set 3, which are the validated neutralizing antibodies, we found a similar pattern, which again validates our sequences. And remarkably, the non-neutralizing sequences from set 2, they were found at a very high rate for COVID active patients, but to a lot lesser extent in COVID recovered patients, and like only to low percentage in the pre-pandemic samples. So we looked closer into the non-neutralizing sequences, and we took all of the sequences which we found in at least one COVID repertoire, and so these are the neutralizing sequences per repertoire, and you see that they are found in both fatal and non-fatal mild disease courses, while the non-neutralizing sequences, they are exclusively found for fatal disease courses. So this indicates that these sequences have like detrimental properties and might be harmful as they do not neutralize, but they bind to the virus. So one reason for the higher likelihood to find SARS-CoV-2 sequences in younger individuals more frequently is like reasons it might be due to the matrix, because we have shown before for T-cell repertoires that there is repertoire restriction in age and cancer, and here we could show the same thing also for the IGH repertoire. Richness and diversity decrease in age, and especially from 60 years onwards, you can see the decreasing richness. And in cancer patients, this effect is even more dramatic. So, in the meantime, our collaborators in the US were working on patients with multisystem inflammatory syndrome called MIS-C, which can occur in adults and children. And this is a syndrome caused by SARS-CoV-2, which presents with fever and hyperinflammation and multi-organ system involvement. It resembles toxic shock syndrome, which is a pathology known for its involvement of superantigens. And T-cell superantigens, they are characterized by the ability to activate T-cells non-specifically. So they bind directly to the MHC, molecule outside of the peptide binding groove and to the T cell receptor without prior processing into peptides. On the T cell receptor side, the superantigen binding is predominantly determined by the shape of the TRBV gene region. So typically superantigens bind to all T cell receptors of the same TRBV gene. And this comes with a cytokine storm, which is observed in some COVID-19 patients. And they did structural modeling of SARS-CoV-2 proteins and complex with the MHC and TCR structures. And then they asked us to look into the repertoires for clues of superantigenic binding. So we subsampled our COVID-19 cohort for patients with especially abnormal cytokine profiles and the ones with more moderate cytokine profiles. And as a nature of superantigen, it binds predominantly to the TRBV gene, and so we did look for shifts in the V gene. And we could find a shift according to the PCA. There was a significant shift in TRBV usage, but not in TRBJ usage as pronounced. So the TRBV genes, which were enriched in the hyperinflammatory group of patients, were TRBV56, V14, V13, and V24-1. And the J genes of these V genes, they did not show skewage, which all pointed to an influence of superantigens. And indeed, our partners, they could model these special V genes in complex with the SARS-CoV-2 and the MHC. And the next step was to sequence their patients because they had a cohort of children with MIS-C, and they had 16 severe and 4 mild samples, and also age-matched febrile controls of children. And this time we did RNA-seq of the whole TCR locus with TRB, TRA, TRG, and TRD. And in average, the mild cases had richer repertoires again, which is in line with the data which we obtained with our adult COVID patients. And again, we looked at the V gene usage and V gene skewage, and we only found a skewing of the repertoire for the TRBV gene. The genes involved were TRBV11-2, and like it had a very dramatic effect. So it was enriched exclusively in severe MIS-C patients, but not in mild patients. And it was like a 7-fold enrichment to over 10% of the repertoire. TRBV24-1 was enriched to a lesser extent, but this gene was also, we have found it before in the adult cohort. That's why this was also interesting. And like for 24-1 and 11-3, there was always like one patient which was especially enriched, and the overall cohort was enriched only mildly. So this is another way of visualizing repertoire, which my PI really fell in love with. And every dot represents one clonal type of the repertoire, and the size of the bubble reflects the fraction of the clone. And here the clones of the same V gene are colored, red is for the TRBV 11-2, and yeah, you can clearly see the cutoff of the severe repertoires with expanded TRBV 11-2. Those expanded repertoire patients, they also had higher cytokine levels for some factors, so especially TNF alpha, interferon gamma, and IL-6 could be correlated with the percentage of TRBV 11-2 usage. Again, speaking for like a supergenic involvement, which triggers the cytokine storm and the TRBV switch. Then we looked into the subgroup of the TRBV 11-2 expanded repertoire versus the MISC samples without expansion. And as superantigenic binding is predominantly mediated by the V beta region outside of the antigen binding site, the CDR3 region should not have any distinct properties. So first we checked for identical clones within the groups, but there were none or not more than you would usually expect. And then we checked, as we did before, the J usage in general and for the TRBV 11-2, but there was no difference. And we analyzed additional properties of the CDR3, so the sequence similarity and the CDR3 amino acid length, but we did not see any patterns. And we also checked with sequence alignments for sequence similarities, and there were not more patches of convergent sequences than normal for the CDR3 region. Only if you take the complete IGH region, you see clusters for the group of expanded MISC, which resembles the sequence of the TRBV 11-2. And finally, we went back to check the V usage of TRA and TRG and TRD as well. And we did find a shift in TRA gene usage, but not in G and D. So the most expanded TRAV gene was 8-4. Again, it was there was one patient with especially high usage, but it was also shifted in the overall cohort. And for that reason, our collaborators modeled the spike protein of SARS-CoV-2 in complex with TRBV 11-2 and TRAV 8-4, and they did get the complex a structure which looked quite relevant to them. Okay, so thank you for your attention, and thanks again to all my co-workers and our collaborators. Open for questions now.
Thank you, Lisa.